Okay, hello everybody. So this is the last lecture of L2 microbiology, as I'm sure you'll be glad to hear. It's also my last lecture of the semester, so of course that's always good news as far as I'm concerned. Alright, so what we're going to be doing is having a closer look at bacteriophages and taking as an example very classical examples, bacteriophage T4 as an example of a lytic phage, and bacteriophage lambda as an example of a temperate or lysogenic phage. So we'll be looking at the lytic and lysogenic replication cycles of bacteriophage. And this will help us understand, how bacteriophages impact human health by transporting virulence factors between bacteria. Once we've done that, we're going to have a look at viral infections in humans. It's a little bit of a general introduction, and then we'll look in a bit more detail at two acute infections that cause serious health problems: influenza and Zaire Ebola virus. And of course, at the end of the lecture, we'll have the final results of the microbe of the week. So I'll try and keep maybe 5 minutes at the end for that. Okay, so bacteriophage, viruses that infect bacteria. They're present in every environment where you find bacteria. Okay, so they're in the soil, they're in our guts, they are in every aquatic environment. And in terms of quantity, okay, of course, the most important source of, or the most important stock of bacteriophage is in the marine environment, in the sea. Now, all the viruses that you can find in seawater are not bacteriophages. They're not all viruses that infect bacteria. You also have viruses that will infect the eukaryotic life in the sea. However, most of the viruses that you can find in seawater are bacteriophages. Okay, so these are the most numerous kind of viruses that you can find on the planet. Now, how many are there? Okay, so let's just have a bit of a guess here. Just say what you think. 1 ml of seawater, how many bacteriophages will you find on average? Marine viruses in total, most of which are bacteriophage. Who says 1? Nobody. Who says 100? Who says 10,000, 10 to the 4 per ml? Got 1, 2, 3, 4, 5, 6. 1 million. Oh, most people. 100 million. 1 person. Okay. Okay, so yeah, 10 to the 6 is about right for the deep ocean water. Okay. If you are out swimming at Pornic and you swallow a mouthful of seawater, you'll be having the seawater on the coastal environment will have more like 100 million bacteriophage per ml, 10 to the 8. So you probably have swallowed 10 to the 9 marine viruses, if that happens to you. Okay, so there are a lot in the sea. And the sea is very big, of course. Okay, so how many viruses or bacteriophage particles are there in all the world's oceans? Okay. So now we're getting some really big numbers here. 10 to the 10, 10 to the 20, 10 to the 30, or 10 to the 40. So who says 10 to the 10? Yeah, obviously wrong. 10 to the 20. Nobody. 10 to the 30. Nobody. 10 to the 40. That's the problem with these like huge powers of 10. You're wrong. Everyone's wrong, right? 10 to the 40 is just phenomenally huge number. I don't know if there are even that many atoms on the planet, to be honest. You know, it's massive, it's huge. There are 10 to the 30 bacteriophage particles. In the world, because, okay, that's because apparently there's about 10 to the 21 liters of seawater on Earth. And most of that water will have about 1 million phage per ml, so 10 to the 9 phage per liter. All right, so you've got 10 to the 9 per liter, 10 to the 21 liters. You've got 10 to the 30 bacteriophage. Now, it's very difficult to try and imagine how big that is, all right, because it's a really big number. So one of the ways to visualize this is to try and think of, you know, how much mass that is. So if you've got, each particle has got about 0.2 femtograms of carbon. So you could easily figure out how many grams that is, or kilograms, but let's try and visualize it and say that's equivalent to how many blue whales, right? Because blue whale is quite big. Okay, so who says one blue whale? 100 whales. All the viruses in the ocean, they put them together, 100 whales. One person. 10,000 whales. Most people. 1 million whales. 100 million whales. You're all out by a factor of 10 to the 4. Right? This number is really big, right? So it's about, let me see if I can read this. Yeah, it's about 75 million blue whales worth of carbon. Okay, this is a huge amount of biomass here. And another way to think about it is, you know, if they're about 100 nanometers long, each one, so if you put them up, if you stack them up end to end, how long would they be? All right, 1 million kilometers. Anybody say 1 million kilometers? 1 light year. 1,000 light years. 10 million light years? Or 1 billion light years? Nobody know? Okay, well, apparently it's about 10 million, that would be 10 million light years long. So that's way bigger than the Milky Way galaxy, and it's 4 times the distance between us and the Andromeda galaxy, apparently, if you put these bacteriophage from end to end. So there's really a huge amount of bacteriophages in the environment. And one thing that is extremely important is this one, okay? The amount of biomass that is tied up in bacteriophage. It's the second largest stock of biomass on the planet after prokaryotes, after bacterial cells. So that has a lot of implications for carbon cycling, the nitrogen cycle, the sulfur cycle, because a lot of this mass is going to be stored in marine viruses. So I'm not a real microbial ecology person, but if you try and think about this, at least in a simplistic way, one of the things that everybody should know about ecology is that every ecosystem starts out with carbon being fixed by primary producers, right? So in the marine ecosystem, this is mostly going to be cyanobacteria, photosynthetic bacteria. So these are going to be eaten by, I don't know, some kind of amoeba or some kind of or eukaryote, microscopic eukaryotes that eat cyanobacteria. Eukaryotes. And then these are going to be eaten up by like krill. Slightly larger organisms, still pretty small, but invertebrates. And then this is going to be eaten by like fish and whales and stuff like that. And then from time to time, these things are going to die, and they'll sediment out and find their way to the bottom of the ocean. And some of this biomass will be lost. Although, in fact, there are communities of microorganisms that live on whale carcasses in the bottom of the ocean. So it's not all lost, really. So it goes to the bottom of the ocean. So if you think of the flow of carbon through a system like this, okay, it starts out with the primary producers, and it moves up to bigger and bigger organisms. So what bacteriophages are doing here is, okay, so these die and they become used by chemoheterotrophs, okay. So what bacteriophages are going to be doing here is they're going to be infecting some of these cells, and they kill them. So that's going to release soluble organic carbon, which can be used by chemoheterotrophic bacteria. Now, some of these bacteria themselves will be infected by phage, and when these bacterial cells get killed, they're going to release a lot of organic carbon. So the net effect of having a huge amount of bacteriophage in the trophic web here is that more organic carbon is going to be recycled at these lower trophic levels. Okay, so this carbon here is not going to become available for feeding bigger organisms, because it carries on being shuttled between bacteria, because phage are infecting bacteria, killing them, releasing the organic carbon into the liquid medium, into the ocean. It can be picked up by other bacteria. Okay, so this is called like the bacteriophage shunt, and the net effect is to keep organic material at the lower trophic levels of the ecosystem. So that's at least on a kind of simple view of what bacteriophage are doing in the marine environment. Okay, so look, there's lots of them. And anywhere you look in any kind of bacterial species, you will find bacteriophage. So there are phage that can infect gram-negatives, gram-positives, mycobacteria, and archaebacteria. Any type of bacterium is got phages that can infect it. And if you look at one particular species, like Escherichia coli, then you can find several different types of bacteriophage that infect one single species of bacterium. So in E. coli, you'll have these tailed phages, like T2, T4, T6. They're almost like strains of the same species. T1, T3, T7, T5. They're all different species. Lambda, M13. So you can easily find like 10 different species of phage that infect one species of bacterium. So that means that there's a huge amount of genetic diversity in viruses, in marine viruses. Well, not just marine viruses, viruses all over the place. So in terms of genetic material and the types of genes that exist, probably most of the diversity in the planet is in phage genomes. It's not in our genomes or the genomes of macroscopic organisms. It's virus genomes that have most of the genetic diversity in the world. And of course, because these can transfer genetic material between bacteria, then of course this is a kind of let's say a kind of storehouse of genetic diversity that can sometimes find its way into cellular organisms. Okay, bacteriophages, what do they look like? So the big classic one is that are tailed bacteriophages called the Caudovirales. Okay, Caudo is for a tail. And these are the ones that are used as the main example because they are the most frequent type of bacteriophage. Okay, so Caudovirales, what do they have? They have double-stranded DNA genome, and they all have a basic structure with an icosahedral head and a helical tail. And within the Caudovirales, so this is an order, there are 3 families, Myoviridae, Cephuviridae, and Podoviridae, that can be differentiated on the basis of their structure. It can be seen in electron microscopy. So Myoviridae, that's these guys. They have a long contractile tail. Cephuviridae, so bacteriophage T4 is an example of a myovirus. Cephuviridae, like bacteriophage lambda, they've got a long flexible tail, not contractile. And Podoviridae, podo means foot, right? So their tail is very, very short, and it's just a kind of foot at one of the vertices of the icosahedral head. So those are the characteristics of the 3 types of Caudovirales. Now, fortunately for us, these physical characteristics are also related to the way these bacteriophage replicate. So Myoviridae mostly are lytic, as are the Podoviridae. Cephuviridae can often be lysogenic. So we'll be having a look at, for example, T4 and lambda, one typical example of the Myoviridae and one typical example of the Cephuviridae. So Caudovirales are the most frequent types of bacteriophages that are found, but that's not the only thing. We're going to be concentrating on those types of viruses, or those types of phage, but there are other things around there. So if you look, you'll find bacteriophages that are filamentous, like this one with a helical capsid. Some of them just icosahedral, just with a head and no tail. And if you look at in some unusual environments, you can find pretty strange morphologies in the bacteriophages. So these examples are all bacteriophage that infect Sulfolobus. Okay, so Sulfolobus is an archibacterium that lives in hot springs. So it's a pH 90 degrees centigrade or something like that. And these virus particles are stable under those conditions. So the external conditions are very, 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 very extreme. That's probably why the structures of the virus particles are very unlike anything you can see anywhere else. And we know relatively little about these viruses compared to, the Caudovirales, where we know all about what is going on. Okay, so for our 2 examples, T4 and lambda, what we want to try and understand is how these bacteriophages, accomplish their replication cycle. Okay, attachment, entry, gene expression, genome replication, assembly, and release. These are the steps that every virus, including bacteriophages, have to accomplish in order to replicate. So attachment. Now, if you're a bacteriophage particle floating around in the sea, and you want to infect a cyanobacterium, you've got to bind specifically to the surface of the cyanobacterium. So attachment is going to be mediated by an interaction between a specific attachment protein on the phage and a receptor on the host cell. So we know some of these. So for T4, the attachment protein is on the end of the tail fiber. Okay, so it's here. And the receptor is outer membrane protein C, which is a porin in the outer membrane. Okay, so if you think about bacterial cell wall structure, T4 infects Escherichia coli. OMPC is in the outer membrane. Okay, so it's available for binding to T4. Something similar goes on with phage lambda. It binds to LAM-B. So LAM-B is a permease for maltose. The J protein, once again, it's on the extremity of the tail fiber here. So J is here. It's going to bind to LAM-B. So, and another example is bacteriophage M13. This time, it's going to bind to something that crosses both the outer membrane and the peptidoglycan layer. That's the fertility pilus, F pilus. So M13 is going to bind on here. It can bind anywhere along the F pilus, and binding of the phage will induce the retraction of this pilus. So that's how the phage gets into the cell here. Okay, it's going to induce the retraction of the pilus, and then the phage will be internalized. Okay, so these are the receptors that are used by bacteriophage infecting gram-negatives. Outer membrane proteins or some kind of structure that is on the outside of the bacterial cell. So how about phage that infect gram-positives? So here's the inner membrane, here's peptidoglycan. The one example I found is a phage called SPP1, which will bind to a protein called UEB. So this is a very large membrane protein which crosses the peptidoglycan layer. So it has part of the structure which is on the outside of the cell, which is going to be available for bacteriophage binding. Okay, so the one thing that's in common here is all phage. Obviously, the receptor has got to be a structure that's on the outside of the bacterial cell. So it's available for binding. Another thing I can say about this is that, okay, if you've got bacteriophage that are infecting a bacterial population and killing those bacteria, this exerts a selection pressure on the bacterial population, okay? So any mutant that arises, for example, that won't express LAM-B anymore, this mutant will become resistant to infection by this phage. So in terms of this one, the consequence would be that if you lose LAM-B, then this bacterium would not be able to use maltose anymore. So there's a kind of cost in terms of resource utilization. But it is possible, okay? So it depends what is more important, using maltose or surviving phage infection. So bacteriophages can have an effect on the biochemistry, the biochemical characteristics of bacteria that are used for identification, okay? Because they can put pressure on what permeases are going to be expressed by the bacterium. Okay, first step is attachment. Sitting around on the outside of the bacterial cell is no good. In order to start an infection, the genetic material of the phage has got to get inside the cell. So there's got to be entry and decapsidation. So here's an electron micrograph of phage T4 on the outside of a cell. What happens is that after binding to OMP-C, there's a conformational change in the tail fiber here. hydrophobic domains of the tail fiber will cross the outer membrane and deliver a phage lysozyme into the periplasmic space here, and that will punch a hole in the peptidoglycan layer. And then that will allow the rest of the tail fiber to contract, cross the inner membrane, and then inject bacterial DNA into the host cell. So that's kind of my cartoon version of this. Now I think I'll try and show you a video here. Okay. Which will show things in more detail. Okay, so in this video, all of the structures are accurate. Okay, so this is the real 3D structure of the bacteriophage T4. So here's the head, here's the tail, here are the tail fibers. So they interact with OMP-C. Then this base plate kind of deploys, and further tail fibers also interact with OMP-C. And this is what's required for, firstly, penetration across the outer membrane. And then these 3 subunits of phage lysozyme will be released. So this is peptidoglycan getting digested here. And then that allows the tail fiber to cross and deliver DNA into the host cell. Okay. So once that occurs, then you have the bacteriophage DNA that arrives in the cytosol of the bacterium. And what needs to happen are these 2 things. You need to have expression of the phage genes and replication of phage DNA. So once the genes are expressed in protein, then these phage proteins can associate with copies of DNA and assemble into infectious phage particles. So in phage T4, how is that organized? Or how is that regulated? Okay. Firstly, okay, after injection of DNA, first thing that's going to happen is that early genes, early messenger RNAs are going to be expressed. So the RNA polymerase of the host cell will bind to the promoters of the early phage genes and start to transcribe them. So you're going to have the host RNA polymerase, which transcribes early mRNAs. Now, what are the functions of these early mRNAs? Okay, they code for phage proteins that are involved in degradation of the host DNA. So there will be a DNase. Replication of the bacteriophage DNA. So there'll be a DNA polymerase. And control of the expression of late genes. So that's now, can anybody see a problem with this? You've got a bacteriophage, it's expressing a DNase and a DNA polymerase at the same time. What is going to happen? Yeah. How can you make, this is going to be like making DNA, and this one is going to be destroying it. So in order for the phage not to destroy its own genome, it's composed of a modified base in DNA. So it incorporates hydroxymethyl cytosine. Wow. Which I'm going to call HMC. So bacteriophage DNA is not attacked by the DNAs. So this is only going to degrade the host cell genome. So another early gene codes for the synthase. So the DNAs will degrade the host genome, and then all the cytosine that is liberated by the action of these DNAs will be converted into hydroxymethylcytosine, which can then be used by the phage DNA polymerase to produce the phage DNA. Okay, so these three things are essential early genes of the phage. GP55 acts as a kind of sigma factor. So this will associate with the other subunits of RNA polymerase, and then this complex is required to transcribe the late genes. Okay, so we've got this. This is the host phage DNAs. Then you have the hydroxymethylcytosine synthase and the phage DNA polymerase, which replicate the DNA, the phage DNA. After that, the polymerase, which associates GP55, will transcribe the late genes. The late genes have two functions. Firstly, they are the structural proteins of the bacteriophage. So this is how you can form the heads and the tails. And secondly, they are the genes that are involved in the host cell lysis. This is the last step. So late genes, structural proteins, and lysozyme to break down the peptidoglycan of the bacterial cell. However, in order for this lysozyme to gain access to the peptidoglycan, it needs to get into the periplasm, right? So it's going to be expressed in the cytosol. Normally, there's no peptidoglycan layer. So there's another protein called a holin, which makes holes in the plasma membrane. So this lysozyme can get out into the periplasm and destroy the peptidoglycan of the host cell. So these two proteins are required for host cell lysis. Okay, so that's how it works. That's how the lytic cycle works for bacteriophage T4. And the important thing to remember is the different roles of the early and the late proteins in this cycle, and particularly why it's important that that late genes aren't expressed all at the same time, okay? Because you have to express these genes at the end of the cycle. Otherwise, as soon as your phage gets in there, it lyses the cell. That's going to be no good because you won't have any infectious particles inside yet. So the way this is controlled is by using this kind of system. So GP55 has got to be expressed first before you get these late genes expressed. And that's very important for the phage. Okay, so that's a lytic cycle. Oh yeah, last thing. Okay, well, I don't really need to know so much about this. Heads and tails are assembled independently, and then they kind of associate. Don't really need to know much about that. Okay, so Myoviridae will generally be lytic, and the example is T4. So Cephuviridae, like phage lambda, can be lytic or lysogenic. That is temperate. So what does that mean? Okay, so let's have a look at this. This is a video microscopy of E. coli, bacterial cells, and they're going to be infected by a bacteriophage, phage lambda, and its genome has been modified to express the green fluorescent protein GFP. So when these cells get infected, they will start to be green, okay? So let's have a look and see if this video is going to run. Oh yeah, maybe. Okay, right, stop right there. Okay, so we've got two infected cells here, right? They're green, this one and that one. So phage have come in, they've entered, you get this, the beginning of phage gene expression. But they're going to have two very different fates, these cells. No, go back. Oh. See if we can go back to me. Okay, let's watch this. This one, oh, it dies. It's been lysed. Whereas this one, this cell just keeps on growing, keeps on dividing, doesn't seem to care that it's been infected. And as it divides, of course, all the daughter cells also express GFP. So they also, all the daughter cells have phage DNA. Okay, so this is a just direct observation of a lytic infection. That's what happened to the cell that was infected up here, and a temperate infection, a lysogenic infection here. So what is going on? All right, so in the cell that has the lytic infection, what you've got is lambda arrives, DNA is injected into the bacterial cell. The first step that's going to happen is it's going to be circularized. That's GFP is being expressed here. Then you have early genes that are involved in DNA replication and late genes involved in DNA production of the bacteriophage structural proteins. And then at the end of the cycle, these phage will be released by lysis of the host cell. So that's what's happening in this cell here, okay? Now how about in this one? What's going on? Well, the first step is the same. The phage attaches on the surface of the bacterium. DNA is injected. It will circularize. But then instead of expressing the genes involved in phage DNA replication, it will express an integrase that integrates the phage genome into the chromosome of the bacterial cell. So this is the definition of lysogeny. Lysogeny is the integration of phage DNA into the host cell chromosome. And once this bacteriophage genome is in the host cell chromosome, it's called a prophage. Now, in the prophage, some phage genes can be expressed, but the late genes that are involved in production of infectious particles and lysis of the host cell will not be expressed, okay? So that's why this bacterium can survive quite happily, even though it's got a phage genome in its chromosome, because the late genes are not expressed during lysogeny. So what is going to happen is every time this bacterium divides, then you'll get daughter cells that all have the prophage, and all of them may express the GFP in this case, but it could be other genes. Okay, so there's two very different outcomes for the host bacterium of a lytic or a lysogenic infection. And it's got to be decided quite quickly, okay, because the choice is made early during the replication cycle, just after circularization of the phage genome. So what is happening? Okay, so once the bacteriophage lambda DNA arrives in the host cell, the first genes that are expressed are these things, N, CRO, C2, and Q. So these proteins are going to control the expression of the rest of the phage genome. And what you need to think about are the roles of these two proteins here, CRO and C2. So these are going to be the master regulators of what happens next. Now, in most cases, C2 will be degraded by the host proteases in the host cell. So what you have left is CRO. Now, CRO will allow the expression of these other genes, N and Q. So altogether, CRO, N, and Q will allow the expression of the genes involved in DNA replication and then the expression of the late genes. So this gives you the lytic cycle. So when C2 is degraded, you get the lytic cycle. However, in some cases, C2 can survive. or C2 can survive, let's say can escape from being degraded. Now, if that happens, then C2 will only allow the expression of two phage genes, the integrase that integrates the phage DNA into the host chromosome, and the C1 gene. And C1 is going to inhibit the expression of everything else, or let's say all of the late genes anyway. So these are two important aspects of lysogeny. The integrase integrates into the host chromosome, and C1 inhibits the expression of the late genes. So it stops the phage from killing the host bacterium. Okay, so what conditions allow C2 to resist degradation? Well, one of them is if the host cell is not growing very fast. Okay, so bacteria that are in the exponential growth phase, they'll have a high metabolic activity, they'll have a high expression of proteases, so lambda which arise will enter a lytic cycle. Bacteria, host bacteria that have poor nutrients, that are not growing very well, or they are at the end of a kind of, they're at the plateau of their growth curve. They're going to have a lower metabolic rate, fewer proteases, so C2 will survive, maybe. Also, if the growth conditions are non-optimal, like lower temperature, protease activity will be lower, so that also will help C2 to resist degradation and will favor lysogeny. The other thing that can happen is if you've got several bacteriophages infect the same cell. So in that case, you'll have a lot of C2 that is expressed, and there'll be too much for the host proteases to degrade. Now, what all these conditions have in common is that they are not good for phage replication if there's a lytic cycle. Think about it like this, okay? Okay, this is your bacterial growth curve here, okay? Lag phase and the exponential phase and a plateau. So if your bacterium is here, right, it's growing really fast. What is going to happen is there's going to be a lot more cells very quickly. So if you have a phage that infects one of these and lyses and produces a whole bunch of other phage, it's very likely that there are going to be a lot of host cells around for this phage, the progeny phage, to infect. So it's good to have a lytic cycle in these conditions when your host cell population is growing, because there will be a lot of hosts. However, if your host cell population is here, the cells are not growing very much. Well, you know, what can happen is you can end up having more phage in the immediate vicinity than you have host bacteria. So if several of these phage infect the same bacterial cell, there's no point in starting a lytic cycle, because the phage that are released, they'll have nobody to infect at the end of it, right? So it's kind of a better strategy for the phage genome to insert into the host cell chromosome and then wait until the host cell starts dividing again, and that way you'll be able to amplify and get more copies of the phage DNA, rather than by lysing, releasing phage out into in the liquid where there are no hosts to reinfect. Okay, so the other thing to remember about this is that lysogeny is a reversible process. Okay, so when the host bacteria are in poor growth conditions, this favors lysogeny and the insertion of the prophage into the host chromosome. When these bacterial cells get stressed, UV irradiation, antibiotics, this kind of thing, that will favor reactivation of a lytic cycle. And then you get a return, then you get expression of the late proteins, excision of the chromosome, and then you get the lytic cycle, which is restarted. So bacteriophage lambda will switch between these two strategies, lytic cycle, lysogenic cycle, and in a kind of logical way that takes into account the ability of the host population to support lytic infection. Okay, so why do we care about this? Well, one of the reasons we care is that, as I hope we saw when we were thinking about pathogenic bacteria, some of the genes that are carried by bacteriophage are virulence factors for pathogenic bacteria. So in these pathogens here, the exotoxins that are, in some cases, are directly responsible for the pathogenicity of the bacteria are carried on prophage. So that's integrated bacteriophage genomes. So what does this mean? This means that bacteriophage or prophages that come from one pathogenic bacterium, if the lytic cycle is reactivated, so you've got your pathogen here, right? Here's this genome. It's got a prophage coding for a toxin, an exotoxin. This guy gets stressed, antibiotics, for example. That will induce the lytic cycle, so you'll get a bunch of bacteriophage. C4 viridae, not my viridae. They can infect commensal bacterium. So then you get a harmless bacterium that has integrated the prophage, coding for the virulence factors. So this then becomes pathogenic. So this is called lysogenic conversion of a commensal bacterium into a pathogen, because the bacteriophage are transferring the virulence factors from one bacterium to another. So this can occur within a species, so from one type of pathogenic E. coli to a commensal E. coli strain, and it can also occur between species. So there are bacteriophage that can transfer virulence factors between Listeria and Staphylococcus, for example. Okay, we're not very interactive today because I have to try and hurry up, but just very quickly, just to make sure everyone's been listening. So what are the functions of the T4 late genes? Hands up. Control the expression of phage genes. Who says that? Control phage DNA replication. Phage structural proteins. Yeah. Lysis of the host cell. Yes. Okay, good. Right, so that's all we need to think about for bacteriophage. Yes. No. DNA in the host chromosome is called the prophage. The whole particle is phage, bacteriophage. Okay. Okay, so the summary here is attachment and entry. How does that work? What are the types of molecules on the bacterial cell that can be used as host cell receptors? And then for lytic lysogenic cycle in T4 and lambda, how are they controlled? What are the different steps in these two different types of cycle? And the thing that is not so easy to understand is why on earth are things so complicated? Why does lambda want to do all this complicated stuff? So why is lysogeny useful for bacteriophages? Why And last thing, the whole bacteriophage summary. Don't forget, there are tons of them. Their importance in microbial ecology is not really clear because it's difficult to study the effects of phage infection on bacterial populations in a real world situation. It's easy to do in a test tube, not so easy to do in an ecosystem. One of the things I didn't talk about, bacteriophage were very important historically as a model system for trying to understand the mechanisms of DNA replication, DNA expression. And a lot of what we know about molecular biology started out by looking at bacteriophage. So things like the genetic code is made up of triplets in a codon with no spaces in between. This was done with by experiments with bacteriophage to try and find out the basic structure of the genetic code. Another thing more recently perhaps is, I don't know, there's one way of changing the genome of organisms in genome engineering in mice, rats, human cell lines. It's done by something called CRISPR-Cas9. And this is a kind of bacterial system for defending against bacteriophages. So research on bacteriophages in the last 10 or 15 years has had a lot of influence on transgenic genome editing technology. Okay, what else to say? So we have, about 30 minutes only to think about viral infections in humans. Okay, so generally speaking, when you talk about, I was ill, I had a virus last week, most people think about acute infections. That is, you're feeling pretty bad, you maybe had a fever, stayed in bed for a couple of days and you got better. So that is what happens in a lot of acute, non-complicated viral infections like rhinovirus, adenovirus, or rotavirus infection. You get infected, you have a peak of virus replication, you have symptoms, and you get better. Now, there are other acute infections which can be very pathogenic. So you can, if you get infected by rabies virus, you probably always die if you don't get treated. So in this case, what is happening is you get infected, you have a peak of virus replication, and your symptoms get worse and worse and worse and worse, and you die. That's also something that happens in about 50% or more of Zaire Ebola virus infections. So you've got a spectrum of outcomes here. So for rhinovirus, probably nobody's going to be killed. And for rabies virus, almost 100%. And then you have lots of cases in between. So influenza, SARS coronavirus, you can have like 0.1%, 1%, 10% of people being killed by these infections in a short space of time. So those are acute infections. Now other virus infections can be persistent. And in persistent infections, you have two types, latent infections and chronic infections. So in a latent infection, what you have is starts out like an acute infection. You get infected, you've got a peak of virus replication, you have symptoms, and then the infection appears to resolve. Symptoms disappear, and you can't detect virus anywhere in the body of this. Well, you can't detect virus in the blood of the person who's infected. However, the virus persists in a latent state somewhere in the body, and then years or decades later, it can be reactivated. And then you have a kind of recurrence of symptoms, virus replication becomes reactivated, symptoms can reappear, and then they disappear again. Then you have these cycles of relapse and remission. So the classic example of latent infections are herpes viruses. So herpes simplex, cytomegalovirus, Epstein-Barr virus, or varicella zoster virus. So if you have shingles, I've forgotten the French name of that, when you are 60 years old or 70, this is the same virus that infected you when you were a kid, when you were like 5 years old in school. And it's been latent for several decades before re-emerging. So that's rather different from chronic infections. In a chronic infection, okay, you've got the primary infection, you have symptoms, and then the symptoms kind of get better. But if you look in the blood of the person, you'll always find evidence of active virus replication. So this virus replication will be ongoing. It's kind of controlled partially by your immune system. But symptoms come back, and as time goes on, things will get worse and worse and worse and worse and can lead to death. So classical examples of that are the hepatitis viruses. Hepatitis B, hepatitis C. You've always got virus replication, and it will have a tendency to get worse and worse. So something like HIV will be in between somewhere. So it's a chronic infection, but there are some latently infected cells. So virus infections are not so simple as you might imagine. Okay, how do you get infected by viruses? Mostly by the mucosa, oral, respiratory, or genital mucosa. Most frequent sites. At the skin, okay, if you get some viruses can be transmitted just by abrasions. So papilloma viruses, they'll cause verrucas on your feet or your fingers. Pox viruses can be transmitted just by the skin. Others can be transmitted by vectors, mosquito bites and ticks. So the skin is pierced by a bite, and then the virus is injected to the cells in the dermis. And of course, plenty of viruses can be transmitted through the blood. Now, all of these mechanisms are a little bit unusual. They're dependent on on technology to a certain extent. So for all of these viruses, there's probably some other kind of mode of transmission that maintains a virus in the population outside of a medical transmission. So for hepatitis B, okay, the way this virus remains at high levels in some populations is last one, mother-to-child transmission, which can be very important. Okay, so at the beginning of the infection, the virus is going to arrive at one of these infection routes. Okay. So what's going to happen afterwards? Well, either you can have a local infection. So in that case, the virus is only going to replicate at the initial site of infection. I mean, if that occurs, then you won't have any virus circulating all around your body in the blood, so you won't have any viremia. And there will be a short incubation time. So the incubation period is the time between infection and symptoms. So that will be less than 1 week for a localized infection. So colds, viral gastroenteritis, this kind of stuff. Now, in a systemic infection, the target organ is distinct from the initial site of infection. Now, to get from the site of infection to the target organ, the virus has got to transit through the lymph or the blood. So you'll always have viremia. This takes time. So you'll have kind of initial several rounds of replication at the site of infection. The virus goes through the blood, affects the target organ. This takes time. So you'll have an incubation period that's longer, at least 1 to 3 weeks. So examples of this are like hepatitis A virus. So the initial site of infection is the gut. The target organ is the liver. Right? Measles. The initial site of infection is the respiratory mucosa. You breathe it in. The target organ where you get the symptoms is the skin. Okay, you get the maculopapular rash. Same thing with chickenpox. HIV. Site of infection is the genital mucosa. The target organ is the lymphoid tissue. So you have some kind of distinction between these two things. Okay, so those are general aspects of viral infections. Now we're going to look at 2 specific examples. Influenza infection and Ebola virus Zaire infection. So 2 acute infections. One is localized and one is systemic. Okay. So influenza is going to be our example of an acute localized infection. So this is the structure of the virus. It's an envelope virus with heliconucleocapsids, look like this. And the genome is unusual because you don't have one molecule of RNA that codes for all the genes in the virus genome. You have a segmented genome. So each virus particle contains 8 segments, and each one of these segments codes for one virus protein. Well, there are a couple of examples, but basically that's the way it works. Now that's going to be one thing that's very important, the segmented nature of the genome. The other thing that you need to know is on the outside of the virus particle, you have 2 viral glycoproteins, hemagglutinin and neuraminidase. The hemagglutinin is the viral attachment glycoprotein. This is the one that binds to the host cells. And if you have antibodies against the hemagglutinin, that will block infection. Okay, so how does pathology work? So these 2 things you've got to remember. Antibodies against hemagglutinin block infection, and you've got the segmented genome. Right, so what happens if you get infected by influenza later this year? Well, you get infected by the respiratory mucosa. Most of the infections will stay up in the upper respiratory tract because of the very non-specific mechanisms we talked about with pathogenic bacteria. The airflow will deposit most droplets in the upper respiratory mucosa, not in the lungs. So your virus arrives up here. 2 days later, you'll have symptoms, on average, fever, myalgia, fatigue, headache. Not necessarily a cough, actually. So this is not always found, but basically fever, muscle fatigue, muscle ache, and fatigue. Then you have, these symptoms will last for 3 to 5 days, maybe 2 weeks or more. If you have a bad flu, you won't feel 100% straight away. So, but that's okay. It's not too serious. Things can become serious if you get viral pneumonia. So the virus comes down into the lungs. That can be that can be life-threatening. Or if you have a bacterial superinfection, you have influenza that infects you, then you get streptococcus pneumonia that infects as well. So these can be 2 very serious complications. Now, for young, healthy people like you, maybe 1/3 or half of the infections of seasonal influenza will be asymptomatic, so you won't even know. You'll be perfectly healthy walking around, coming into lectures and everything else. But people who are elderly or young infants will have a higher frequency of these complications. And overall, 1 year to the next, you can have about a 0.1% mortality rate. For seasonal influenza. That's not very high, but for a virus that can infect millions of people every year, something that is a serious cause of morbidity and mortality in elderly people in particular. So that's why people over the age of 65 should be vaccinated every year. And this one, because you have asymptomatic infections in young adults, is why people who are involved in the care of elderly people or patients in hospital should also be vaccinated every year, because it will prevent these asymptomatic infections, because these people will be infectious. And if you come into contact with someone who's very susceptible, you can infect them and it can be dangerous. So that's why vaccination is important. Now, the purpose of vaccination, or the objective, is to generate antibodies against the hemagglutinin protein, and that will block the infection. However, the problem is this has got an RNA genome, and it's very variable. So from 1 year to the next, the hemagglutinin protein on the surface of the virus mutates. This is what's called antigenic drift. So that means that the vaccine has got to change from year to year. So this graphic just shows you the different strains of virus that have been used to make the vaccine for influenza up until 2001, 2002. So the one that seems to change the most is the H3N2 strain. So this one had to be changed very, very frequently. And this is a big problem in terms of the efficacy of influenza vaccination, because the decision of what strain to put in the vaccine has got to be made early in the spring, right? So the vaccine that is available now, the World Health Organization working group decided what strain to put in it earlier this year. And it's a prediction, all right, about what is the strain that's going to be circulating in the Northern Hemisphere. So in the years when the prediction is good, the vaccine is effective. In the years when the prediction is bad, the circulating virus is not the same as the vaccine strain. So you can be vaccinated and you won't be protected. Okay, that's because mutations accumulate in the hemagglutinin protein. That's what happened last year. So in the UK, probably tens of thousands of excess deaths because of the poor efficiency of the seasonal influenza vaccine in elderly people. Probably also in France. So that's one of the problems with influenza. The other one that is that you can get something called antigenic shift. So this occurs when you've got two different influenza viruses that infect the same person or the same animal. And in that one animal, two different viruses infect the same cell. Now, because the genome is segmented, okay, inside this infected cell, you'll have genome segments from each of these two viruses. And then when this cell releases progeny virus, some of them can contain some of the genome segments from virus A and others from virus B. So this gives you a kind of mosaic virus through reassortment of the two virus genomes. And that can be very serious because it can be one of the sources of pandemic viruses. Now, pandemic influenza virus is a virus that suddenly appears somewhere in the world that hadn't been circulating in the human population before. It will have a hemagglutinin protein that's very different, and no one will have immunity against it. So it can infect a lot of people. So the last one was the H1N1 Mexican flu in 2009. Fortunately, it was not very pathogenic, so it didn't cause a lot of deaths. But over the 20th century, there have been several pandemic viruses that have been pretty pandemic. So this one, the Hong Kong flu, caused a lot of deaths all over the world. And the big daddy of all pandemic flu viruses was the Spanish influenza. Probably caused 25 to 50 million deaths over the course of 2 years when it emerged. So there are two causes of pandemic influenza viruses. One of them is this mosaic, okay, the reassortment of genome segments from an avian virus to a human virus. So you get a kind of mixture. And the other one is direct transmission of an avian virus to humans. So that's what happened with the Spanish influenza. So that's why avian influenza is under worldwide surveillance. And what's the news story this morning with avian influenza? Anybody hear about it? We have H5N1, highly pathogenic influenza, avian influenza in the Dordogne. So there are control measures in the farms around that affected farm. Okay, we have time for about 5 minutes on Ebola virus infection. Going to be a bit short here. Okay. Very quickly, Ebola virus, okay? Also, negative strand RNA virus. This time doesn't have a segmented genome. All the genes are on the same RNA molecule. It's an envelope virus with a helical nuclear capsid. It's got one envelope glycoprotein. This is the G protein, all right? So in this case, antibodies against the G protein confer immunity. Okay? So for influenza, it's antibodies against hemagglutinin. For Ebola virus, it's antibodies against the G protein. Now, we're quite lucky because the virus doesn't show very much genetic variability from one outbreak to the next. So it's quite genetically very stable. So that means that vaccination could be effective. So there were several candidate vaccines under development. And earlier this year, there were very good results with this one. So it seems like we now have an effective vaccine against this virus, which is very good news. So this was in here because last year there was a very, very severe Ebola virus outbreak in Guinea, Liberia, and Sierra Leone. It infected tens of thousands of people and killed 10 to 20,000 people. Now, all previous Ebola virus outbreaks have been very small, maybe 100 or 200 people infected at the most. And that's because this is not a virus that circulates in the human population. So basically, Ebola viruses infect bats. From time to time, these bats can transmit infections to other wild animals. And if these are hunted for food, then there can be transmission of the virus into the human population while people are cutting up the meat and preparing it and getting it ready to cook. Once you have transmission into the human population, you can have limited transmission from one individual to the next. And really, it was a huge surprise for everyone last year when we saw when there were transmission chains infecting hundreds and thousands of people in these 3 West African countries. So what happens to you if you get infected by Ebola virus? It's a systemic infection, okay? You have an asymptomatic phase, an incubation period of up to 21 days, up to 3 weeks. So after that, then you'll have early symptoms, which are very non-specific. Fever, fatigue, myalgia, headache, vomiting, diarrhea. Could be lots of different things. It's not very specific. But what is going to happen is that after 2 or 3 days like that, you'll have a worse and worse fever and start to have a hemorrhagic syndrome. So you are bleeding from mouth, nose, eyes, the anus, and in the end, you'll lose your consciousness, you'll pass out, and you'll have internal hemorrhage and die. And this will happen to maybe 50% of the people who are infected, 50%, 70%. So it's extremely dangerous. And you only have like 8 days between the onset of symptoms. That's here. You think you've got the flu, and then 1 week later, you're dead. So it's probably, it's really dramatic and very, very serious. So what is happening here? Okay, during the asymptomatic phase, you've got 2 weeks, you're infected, but you don't have any symptoms. Well, what is happening is the virus is disseminating all through your body, but your immune system has just gone to sleep, doesn't detect anything. Then later on in the symptomatic phase, suddenly your immune system wakes up, oh my God, we've got virus everywhere. And so you have a huge activation of monocytes and macrophages all through your body. So what are they going to do? They're going to secrete a whole bunch of cytokines that induce fever, so you've got a very, very high fever, and other factors that will have an effect on the permeability of endothelial cells in your blood system, the expression of molecules that favor coagulation. So you have diffuse intravascular coagulation. And this will use up all of the coagulation factors in your blood. So then afterwards you start bleeding and you die. Okay, so you have a very severe, it's very similar to toxic shock caused by endotoxin. So the reasons why people die are quite similar between toxic shock and an Ebola virus infection. All right. Last words about this. How do you catch Ebola virus? Well, you have to have direct contact through to some contaminated material. So contaminated body fluids are tears, saliva, sweat, blood, feces, vomit. All of these can transmit infection. That's why you have to have people with these massive suits and quite severe rules about how you put it on, how you take it off to avoid the doctors and nurses getting infected. Right, not all people who are infected are very contagious. During the asymptomatic phase, there have been almost no incidents of people getting infected from an asymptomatic infected person. So no transmission during the asymptomatic phase. People become contagious once there are symptoms, and then as symptoms become more severe, people get more contagious because there's more and more virus around. And they're extruding it through their blood, their sweat, and other bodily fluids. One of the things that was very important was corpses remain infectious. So in West Africa, when people were killed by this virus, and they got before they were buried, there was a funeral. The relatives, the family would come up and hug the dead person to say goodbye, and this was very dangerous because it was a very important source of transmission of the infection. So one of the things that was able to control these transmissions were procedures for safe and respectful burial, that people could so people could bury their loved ones with dignity, but not in a dangerous way. Okay, I think we're just about running out of time here, so we'll stop there. Things to remember, okay? Diversity of viral infections, acute, persistent, local, systemic. For each one, you've got a kind of diversity of clinical outcomes. Even for seasonal influenza, the death rate is about 0.1%, and your outcome of an influenza infection goes from asymptomatic for a young, healthy person, 30 to 50% of the time, to a very severe infection and with a risk of death for an old person or a young child with a tendency for asthma. What can we say about this? In both of these cases, there's a kind of link between transmission from an animal reservoir to the very highly pathogenic forms. Okay, so in some sense, there's a kind of mismatch between the virus and the host immune system because it's a zoonosis, an animal virus being transmitted to humans, and this can be very, very dangerous. Okay, so we're going to wrap up there. We have 2 minutes, 3 minutes for microbe of the week. Because, as I said before, microbe of the week as a prize. So here are the final results. Oh, someone can take a picture when I give this, when we give these out. That'd be nice. Okay, as long as people don't object to being on my Facebook page. Right, so ça va. Okay, so here are the final. Oh, here were the final results. Okay, right. So we have number 1, Alexandre. Let's have it in reverse order. So there's a tie for second place here between Harriet and Angelique. So a lot of very good answers all the way through the system. So you've got a tie for second place. So there are going to be 3 prizes, right? So come up here. Number 1, okay, was fast out of the starting block. So the first 3 answers, Alexandre Bruno. He slowed down in the home straight, but he was just too far ahead. It was just impossible to catch him. So first prize, Alexandre Bruno. So come up, step up, please, everybody. Yes, yes. So come on. So the first 3, step up and receive your prizes. Okay, so it's microbiology, and we studied fermentation. So I thought it would be appropriate to have some fermentation products in the prizes. So third prize, some choucroute. Okay, cool. Okay, please enjoy your prize. Second prize, what do we compare choucroute to? Kimchi, yeah, okay. Here is some kimchi. Be careful when you open this, all right? Might be a bit of a surprise. And first prize for Mr. Bruno. You are. Okay, so thank you for everyone who sent in answers for Microbe of the Week, and good luck for your exams.